Thank you. I got so involved thinking about what Paul said, I almost forgot I was speaking next, which I guess is a compliment to Paul. So I'm going to talk about an effort that's been ongoing at EPA for almost 4 years now, thinking about how to advance the next generation of risk assessment, which we fondly call NexGen. And it's essentially just looking at recent advances in science and how can we use this information to better inform risk assessment. I'm going to say we throughout this whole conversation, but I want everybody to be clear it's the royal we. Individuals from all of these organizations participated, and I did very little real work in the project. So I want to start out, this is a timeline of what we did, and we started with these influential reports and looking at the recommendations of those reports. I know the Academy sometimes thinks that EPA actually doesn't listen to what they say, but it's not true. So we started here, and one of the things that was common to those reports was a recommendation that prototypes that could engender movement from strategy to practical application of new kinds of data in risk assessment was a common recommendation. So that was an important focus of the NexGen project. The other thing we did was we met with decision makers early on, because one of the things we paid attention to was something that John had raised about these fit for purpose or right, I can't remember what the term you used that I like, right fit sort of risk, right size risk assessments, thank you. You know, that decision makers have different needs for information, and that you appropriately could save a lot of time and energy by targeting your assessment to what the decision needs are. So after we spent some time trying to clarify what decision makers really needed and how we might serve those needs, we began to look at information that was available and pick out prototypes. There was a meeting in November 2010 with experts to talk about and refine the prototype concepts. And then in February of 2011, we had a public meeting to communicate both what we intended to do and to take comment on the plan, as well as advice on how to communicate during the process, as well as the results of the effort. And so to some extent, the final report is kind of a fulfillment of promises that we made at this public dialogue conference. The other thing I wanted to point out was, very independently, the National Academy began a series of workshops on emerging science, which proved to be this invaluable forum for discussing of ideas, both listening to other people's ideas as well as presenting ideas. I think that all of the team leaders for the different prototypes at one time or another were asked to present at one of the workshops. One of the things that came out of the public dialogue conference was this obvious split, or not split, but continuum of what the kinds of information that stakeholders want and the technical knowledge that stakeholders could bring to the process. So one of the things we decided to do is to kind of have a multi-level reporting out, if that makes any sense. So we decided that there was a need for a series of technical papers that were primarily targeted to the scientific community. There are approximately 40 papers that either have already been published or will soon be published that are a product of NexGen. And then we wrote kind of a summary report that was intended to be user-friendly. And there's an executive summary, which is the real user-friendly, and then there's the document itself, which we were partially successful, I think, at being user-friendly. That report is now an external review draft. It's in peer review now. And public comment period is open till January the 14th, I believe. So if you Google EPA and NexGen, you can pull up the report as well as instructions on how to provide comments on the document. So for the rest of the talk, what I'm going to do is highlight a couple of important messages, I think, that are in large part responsive to some of the NAS recommendations. So we we kind of developed three decision contexts or fit for purpose sort of categories for risk assessment. This doesn't mean there are only going to be three kinds of risk assessments, but for illustrative purposes, we tried to look at three kinds of decision making. As you go from top to bottom, as you go from top to bottom, what one sees is part of the decision context is a greater and greater number of chemicals that the decision maker has to consider. So it's something at the top level might be what we're familiar with the IRIS assessments or the criteria document assessments now, where you're dealing with relatively few chemicals but lots and lots of data. Limited scope assessments might be something like dealing with Superfund chemicals, where you might have hundreds to thousands, a few thousand chemicals that a risk manager would have to make some kind of decisions about. And then lastly, the high throughput ranking and screening type risk assessment, where what you're really talking about are thousands, potentially tens of thousands or hundreds of thousands of chemicals that one might want some information on. Because Richard Judson is going to very ably talk about this in the next session, I'm not going to talk about it here, but rather I'm going to focus more on this for reasons I'll go into in a minute. This section is going to get kind of short shrift, but the things that were done here were knowledge mining. In this example, as a collaboration between NIEHS, EPA, and Stanford, we did a lot of knowledge mining to look for associations between diabetes and obesity. I think knowledge mining of big data sets is like just a critical area for future development. And then we had two prototypes that were short duration exposure, in vivo exposure kind of paradigms. One that looked at alternative species and the other one that looked at mammalian species. And so this was led by Ed Perkins at the Army Corps of Engineers, and Rusty Thomas led this one, who used to be at Hamner and now is at EPA. So what I'm going to talk about more is this group of prototypes. I got a lot of grief where people would say, why in the heck did you look at benzene, ozone, and PAHs? We know everything we ever needed to know about them. Why use those as prototypes? And the reason was exactly how much we knew about those chemicals. The criteria for these, what I called proof of concept assessments, were in vivo human exposures at environmental concentrations where we had a good handle on exposure, and hopefully we had a good handle on exposure dose. There are not many data sets that are like that. We also wanted to have data that were traditional endpoints as well as molecular endpoints were collected in the same study. What we intended to do was iterate back and forth between the new types of data, the omic data, the cell biology data, as well as the traditional data, in hopes of both demonstrating proof of concept of what we could do, understand what we couldn't do, sort out what information was valuable, as well as begin to develop decision rules that would help us use new types of data appropriately to get the, quote, right answer. So what you see, for those of you who don't spend your time in molecular biology, what you see on the right-hand side are essentially diagrams that show relationships between genes and gene activities as underlying mechanisms of disease. Different people call these different things. They get called signatures, fingerprints, network diagram, nodes and edges diagrams. They get called a bunch of different things, and people use different programs for mapping out the diagrams. But essentially, all of these are maps or diagrams of molecular mechanisms underlying disease. And one of the things that we could identify from looking at these either molecular epidemiology or molecular clinical studies were that you could in fact identify molecular patterns that were predictive of specific hazard and risk, dose response. It's considerably easier to do hazard than it is dose response, but it can be done particularly if you have a good handle on your exposure dose parameter. The other thing that we did was, even though we focused primarily on these big chemicals that we know a lot, benzene, ozone, and PAHs, we also tried to go to look at data from chemicals that produce similar outcomes. And one of the things that we found useful was that chemicals with the same outcomes appear to impact the same networks. And the reason that this is important, that if we can identify these underlying molecular patterns of disease, these patterns can help us characterize data scant chemicals as well as cumulative risks, as well as human variability. I want to make clear that in no way am I proposing that for each risk assessment we should go out and gather the kind of data we used here. That will simply slow the process down logarithmically. But rather, these were used as proof of concept sort of examples. And I'll give you some examples of how I think these kinds of data can help us in terms of sorting through lots of chemicals more rapidly. So if you look at, this is a KEGG diagram, which is the Kyoto Encyclopedia for Genes and Genomes, for those of you who don't spend your time looking at these things. And these have been identified primarily from de novo disease, or essentially disease of unknown etiology, of genes and pathways that are involved in acute myeloid leukemia. So if you go then and you look at not only benzene, but other examples of leukemogens, as well as non-chemical risk factors, you can see how these tend to alter genes and pathways within this same network. And one of the ways I think this is important in terms of screening other chemicals, this is all in vivo human data, but if you had simply in vitro data, say you're a chemical that's trying to evaluate what chemicals you might want to use in a process, if you look at in vitro data for a chemical that's never been tested in vivo, and it tends to disrupt or affect these same genes or processes, that would increase my concern for the chemical as a leukemogen. It doesn't mean that it is, but it would make me more concerned about that chemical than a chemical perhaps that did not look like this. Another aspect, you can see easily how you can begin to use these kind of maps to evaluate the effects of mixtures or in other environmental factors in a cumulative risk kind of way. You know, obviously chemicals that interact in the same network are more likely to, I think, sort of interact in terms of the risk of that disease than chemicals that don't interact in the same network. And then the last point I wanted to make is that from this robust sort of human disease database, we know that there are genetic variations in many of these genes, and those genetic variants impact risk. They impact the incidence of leukemia as well as the prognosis for leukemia. And I'll give you an example. This is a study looking at relative risks where people either had or did not have a particular polymorphism. This paper looked at several polymorphisms, but this is just one variant in the human population. And you can see how there's an appreciable impact on the odds ratio for leukemia. One of the things that could be done, since it is to map the multiple variants that are significant subpopulations in the United States or whatever population you were interested in, you could in that way begin to understand the genetic contribution to human variability. So before my enthusiasm about all the things that we can do leads us astray, I wanted to mention that the other thing we learned is sort of the significant challenges. There's lots of unusable data out there. If we had known then what we know now, a lot of the studies we've done we wouldn't have done in the same way. So systematic review and adherence to best practices of data that gets used in risk assessment is going to be critical. Consideration of variability is crucial, that the signal to noise ratio can eat you alive in these studies. a whole new set of uncertainties need to be described that we haven't spent the last 30 years discussing. And that it's really critical that these omic changes be embedded in a mechanistic network or context, because all of us, when we ate breakfast, we got up, we changed what was going on sort of omically in our body. And to be able to separate out what are just normal biology from disease biology requires that you put these things in a sort of a more of a network context. And then the last 2 points I want to make are sort of for the future. One of the things you don't see are studies that in a single study look at multiple omics. People tend to look at proteomics or genomics or transcriptomics, but you don't see these kind of tools brought together in single studies. And we know enough to know that all these things are probably going on with chemically induced alterations, but the studies that you'd like to look at omics in an integrated fashion are pretty rare. And the last point I wanted to make was that all of this is a snapshot in time. The studies we tend to get are a snapshot of the biology. My colleague Lyle Burgoon says currently what science does, it gives you a roadmap of what's going on in terms of a disease process. And what you're really interested in is traffic flow, right? And we're kind of not there yet. We're really interested in information flow in the organism. And then the last point I want to make is we're still very stovepiped in this modern era. And I think by integrating what we can know from human disease and human genetics that come out of the disease, just the pure study of disease in the absence of chemical exposure, as well as the more experimental data from chemical exposures, and integrating those two is going to give us the best information that will allow us to screen new chemicals more rapidly using new omic methodologies. And I think using a combined approach, then you can do a variety of things. And then the last thing I'd like to point out is Chris Portier presented a version of this slide at our kickoff meeting in 2010, which just proves that Chris usually has some of my best ideas about 2 or 3 years before I do. But so, thank you. Oh, and my acknowledgement slide. So many people helped on this that I wanted to acknowledge them. Thank you.
And just a quick announcement before our last speaker comes up, and that is, if anyone is a speaker, to please not bring their cell phone to the podium or bring their cell phone when we sit at the panel table. We're getting some feedback, and so we want to make sure people who are listening via web can get that. So we're moving a little bit forward now to actually something that's in progress. And our last speaker is Marilee Shelton Davenport, and she's a senior program officer with the Board of Life Sciences for the NRC, where she's worked on a variety of biology projects since 1999. She's currently the director of the Standing Committee on the Use of Emerging Science for Environmental Health Decisions, which facilitates discussions on the scientific advances for the identification, quantification, and control of environmental impacts on human health. She's also the study director on a consensus committee, and we're going to hear about that committee today, the design and evaluation of a safer chemical substitutions, a framework to inform government and industry decisions. Thank you, Marilee.